It's every biomedical investigator's dream to contribute toward a therapy that more effectively treats disease. That's doubly true if it's a disease that, if it's a treatment that can save a life. And it's even more true if the life that you save is that of a kid, a child. It's my great honor and pleasure to introduce today today's speaker and my long-term friend, Carl June, who has lived that dream. I'm going to quickly dispense with some formalities, and I've written my introduction because I could spend the whole hour talking about Carl, but I don't think you're here to hear me. But you'll notice a theme. Carl graduated at the top of his class from the U.S. Naval Academy in Annapolis. Then he went to Baylor, where he graduated with AOA honors and as the Michael DeBakey Scholar for Outstanding Performance. Then he achieved the highest board exam score in the state of Texas for his qualifying exams. Then he won a one-year World Health Organization research fellowship, which he did in Geneva. Then he completed his internship and residency also at the National Naval Medical Center and was selected to serve as chief resident. And then he completed a 2-year fellowship at the University of Washington's Fred Hutchinson Cancer Research Center with future Nobel laureate at the time, E. Donald Thomas. The point that I think you are picking up is that Carl strives for and achieves excellence in all things. For example, he has had a long-standing interest in physical fitness, with cycling being his favorite form of torture. And I can attest to the fact I'm a victim of his torture on many long rides that we took from Rockville, Maryland to White's Ferry on Saturday or Sunday mornings, or to Sugarloaf Mountain to climb that. He has one speed, and it's fast, always fast. He launched his independent research career at the Uniformed Services University in Bethesda in 1986. And for 13 years there, he conducted basic research into understanding T lymphocyte function. I'm showing you this slide. I had been at the Navy Medical Research Institute for about 2 years. That's me when I was younger. Here's Carl on Funny Hat Day. This was his idea, wear Funny Hat Day. What the picture doesn't show is the incredible hard work that we did and had fun doing it. We had some quirky team members, I think Carl would agree. We had some bad experiences, like losing a huge liquid nitrogen-filled freezer stocked with irreplaceable supplies that thawed. Everything melted and thawed. There were political struggles, and unfortunately, we lost some dear friends and loved ones too often from cancer. But the camaraderie was unbelievable, and the energy of the place was unbelievable, and just the thrill of discovery was unbelievable. In 1999, Carl moved to the University of Pennsylvania, where he is the Richard Vague Professor of Immunotherapy, and he directs the Center for Cellular Immunotherapies at Perelman School of Medicine, but also the Parker Institute for Cancer Immunotherapy, which is a joint venture with, I believe, Stanford and UCSF. Is that right, Carl? He's published over 480 peer-reviewed manuscripts in the highest quality journals, has scores of patents, and has won many major awards, including the Dexter Conrad Award, which is the highest award for scientific achievement given by the Office of Naval Research, when most people that get those awards are submarine engineers and things like that. He was elected to the American Association of Physicians. He's won the Steinman Award from the American Association for Immunology, the University of Pennsylvania's Osler Award, the E. Donald Thomas Award from the American Society of Blood and Bone Marrow Research, and the American Society of Clinical Investigations Harrington Prize. And that's just a smattering of the awards, and I'm sure many more will follow, including, I think there's one giving in Stockholm. I wouldn't be surprised. Since I see many young people here, I'm just going to conclude with things that I've observed from Carl that I say are the secret to success. And so if you do these things, you're pretty likely to succeed. One is you need to be wicked smart. 2, it helps if you outwork everyone. And I've never known someone that works harder or longer hours than Carl, not one. He's the top. And it's best to be character-driven, laser-focused, and very resilient. Carl doesn't wallow in it, but I'm aware of several personal losses that he's suffered, and I've always been astounded with his rebound. It's not that he doesn't feel the blows, because he does, but he gets back up again pretty quickly. And the primary lesson I've learned from Carl is this: the surest bet to achieving a great and important goal is to pick a huge one, worthy of your life's work. Focus on that goal, demanding excellence of yourself and all around you in all the things. And with persistence, teamwork, and resolve, you might just hit that goal. I have one more slide. Among Carl's many awards, I'm sure this is considered a minor one for him, but we were the co-recipients of the first inaugural Frank Brown Berry Prize in Federal Medicine back in 1997 for work that Carl initiated long before I came. He's had other important and interesting experiences. This was his Christmas card about 2 or 3 years ago. But I think the only award that really matters, and I'm sure Carl would agree, was this Christmas card. And each one of those young people you see there had been given a terminal diagnosis until, and the family got the word, I'm sorry, there's nothing more we can offer, until they got CAR T cells. And now each of the people in that picture are cancer-free. So Carl, I see that I emoted in you. Please welcome my friend, Carl June.
Well, thank you very much, and I want to thank my first wife. I lost her to ovarian cancer, and Dave took care of me to get me through that. So thank you. So disclosures are Novartis, and has sponsored much of our work, and I'm a scientific founder of a pen spin-out called Immunity that is also making CAR T cells. So my theme today is cancer immunotherapy, where the ideas have been over 100 years. You know, it began with Paul Ehrlich, first seeing what was called lymphocytes back on microscope slides surrounding tumors, and he made the idea back then in the 1860s that there could be magic bullets. This before we even knew what antibodies really were. And then, but it never really worked until 2010. And that's when checkpoint therapies were first approved in melanoma for anti-CTLA-4. And that led to the Nobel Prize last fall to Jim Allison and Tatsuko Hanjo for PD-1 and CTLA-4 checkpoint therapy, which is probably a $50 billion industry at this point. The other main immunotherapy is CAR T cells that has come out, and that's what I'll talk about. But they both enhance the immune system and overcome tolerance. So tolerance, when I went to medical school and was not well known, we thought that the thymus deleted autoreactive T cells. The forms of central tolerance are shown here as A, B, and C. Basically killing T cells at the thymic level and making them not respond to your own organs so that you don't get graft-versus-host disease. And in humans, it's turned out there's many examples of peripheral tolerance. And basically cancer in every single way has phenocopied some of these as a way to avoid being destroyed by the immune system. The main thing that checkpoint therapies do is overcome exhaustion, a new state of T cell function, which is where the T cells are alive but really don't proliferate. And checkpoints can help overcome that. What CAR T cells do is overcome tolerance by the insertion of an antibody, and it can directly bind to antigens at high affinity on tumor cells. So CAR T cells didn't just happen. The first issue was making a T cell that's not HLA restricted, because normal T cells work through transplantation antigens. And that was first done by this group where they grafted antibodies onto a T cell receptor and showed then you could have HLA-independent recognition of a target, initially haptens. And then Art Weiss did a seminal experiment where he grafted the extracellular domain of CD4 onto the T cell receptor zeta chain. This T cell receptor has 7 or 8 chains, And he showed that this so-called first-generation CAR that had just the zeta chain of the T-cell receptor could replicate most of what the natural T-cell receptor does, where it has 8 chains. And then investigators showed you could use antibodies or other molecules and redirect at will to any target that's on the outside of a cell. And it could be a healthy cell or it could be a tumor cell. And then these initial just were signaling domains of just the zeta chain. Then other domains such as 4-1BB, which I'll talk about, and CD28 were added on, and these were called second-generation CARs, and they are now what have become FDA approved. And something that's not well known is that the initial CAR development was actually done in biotech, and it was done by Helene Finney and Margot Roberts. So Helene was at Celtech, and she was working on CAR designs, and then Kristen Hagee is a long-term collaborator of mine. She was at Cell Genesis along with Margot Roberts. They developed a CD28 CAR, and Margot's now at Kite doing with their CAR called Yescarta, and Kristen Hagee is close to getting FDA approval of a CAR for myeloma that targets BCMA. Well, what happened is there was a big catastrophe really in gene therapy in 1999, and the industry left. They decided it would not be possible to ever get commercial development of cell and gene therapy, and the rest of it then took over and was done in academia, and that's where my story continues. So I initially worked with Kristen Hagee at Cell Genesis, and we did the first CAR trials in actually HIV, and it was a CAR called CD4 Zeta, and then that had safety and some antiviral efficacy. And then I did the first trial with her where we did the cell manufacturing. She was still at Cell Genesis in patients with colorectal cancer targeting an antigen called TAG72. It didn't work at all. And then these 2 trials also failed with first-generation CARs, one at the NCI and the other in Amsterdam. So then second-generation CARs became developed where either the CD28 signaling domain was added onto the Zeta or the 4-BB domain. And this led to FDA approval of Yescarta by Kite Gilead in 2017 and Kymriah by Novartis in earlier that year. So the CAR that we got developed in my lab and then became Kymriah was developed by Richard Carroll, a postdoc in my lab, started in the Navy with Dave Harlan. He unfortunately died early of pancreatic cancer at age 50, and then carried on by Michael Malone, a postdoc in my lab. And so what Mike did was a quantitative experiment to ask how you could make CAR T cells survive better. And so he injected immunodeficient mice with a 1-to-1 mix of CARs, either first generation or second generation, and then waited 6 months in these mice and found which CARs survived. And if they survived equally, they'd be on this red line. And what you can see on this log scale is that there's a big bias to CARs that had 4-1BB signaling domains in them. And that's why we use that in our clinical trials. And, the preclinical data shown in 2009. Our first patient was treated in 2010. But we then found out we were doing something we hadn't even suspected with a CAR with that signaling domain. And that is we made CARs targeting the same CD19 molecule and either had CD28 as a co-stimulatory domain or 4-1BB, and then activated them by adding in leukemia cells, which makes the CAR cells proliferate. And if you looked at them, you could visually see the difference in the cells. These cells turn the media orange very rapidly, which means they're making lactate and buffering the phenol red. And it turned out they're just addicted to sugar. They work on glycolysis, so-called Warburg effect of aerobic glycolysis. These cells didn't turn the media orange, and what they use is fatty acid oxidation. And if you do electron microscopy, they're full of mitochondria. So we found you could not only change the specificity of what a CAR T cell will bind to by redirecting the antibody on the outside, but you can change its metabolic fate by selecting different signaling domains. So you can have cells that will live a short time or a lifetime, depending on how you, and you may want both, and depending on how you design these signaling domains. So coming back, CAR T cell therapy now is something that's really been disruptive to the pharmaceutical industry because it's not just a drug that you buy. It's a process, and it's most related to a transplant. And so that begins with, right now, using autologous T cells that are obtained by phlebotomy from the patient. And then there's an ex vivo growth in the lab, which initially for our initial trials was 10 to 12 days. We now have a 3-day manufacturing process. The cells are then frozen in infusion-ready DMSO, and then frozen until the patient's ready for infusion. And then there's a 37-degree water bath in the room, and the cells are thawed and just infused IV in general to patients. So those are autologous cells. There's also a lot of research now on using allogeneic cells, coming either from healthy donors, iPSCs, cord blood that's normally discarded, and so on. So in the end, we're going to have two kinds of products, those that are off the shelf and made from someone else's and can be given immediately, and then others that take longer to manufacture, and it's more expensive, but they can live a lot longer in the patient, the autologous CAR cells. So this compares the two commercially available CARs. Their UCN names are shown here. This is Tisagen Laclucio, which is Kymriah, and this is the Gilead CAR. So we use different vectors to make them. One's a lentivirus, and there's a mouse gamma retroviruses, different promoters. The same binder derived from a mouse hybridoma that binds CD19, different signaling domains, and other differences in design. And these now we know survive. Actually, in our first patient treated, still has CAR cells 8 years later circulating in him. These survive less than usually a month in the patient, and we think that's primarily due to the signaling domain. So, and I think you want both short survived cells that are really good killer cells and other cells long-term immunosurveillance. So we first treated CLL patients in 2010 in July with that CAR, and then reported it, our first 3 patients, 2011, and that really triggered a storm of interest because we had all 3 patients respond, as I'll describe. Then we began treating pediatric leukemia and reported acute leukemia. So this is chronic leukemia and pediatric leukemia. Reported that in 2013. And we've treated every kind of B-cell malignancy, and every one of them has responded, all using, in this case, autologous CARs. And leading to FDA approval in August 2017. So the first patient we treated had a P53 mutant chemo-refractory CLL. His name is Bill Ludwig. He was a retired Marine. And I can say his name because he was on the New York Times, and he gave up his HIPAA protection. When we got FDA approval, he's still getting infusions. So here he is getting his CAR infusion. This is 7 years later, and when he's in an infusion chair, he's getting gamma globulin. It's because he has an acquired B-cell deficiency from the CARs, which also target normal B cells. He has no leukemia, though, and what he said was, I was patient number 1 with CART-19, and all I got was this T-shirt. So, and I literally got an email from him about 3 weeks ago. It was his anniversary now from the next year on, and he's still leukemia-free. So this is what we learned out of those patients. The cells are genetically marked, so you can count them in the patients. And if you look in Wikipedia, a kilogram of tumor is a billion, 1,000 billion cells, 10 to the 12th. And so our first 3 patients had 3 to 7 pounds of tumor. So the spleens, bone marrows packed, et cetera. And what we found was the proliferation of the CAR cells correlated with elimination of the tumor. And basically at the time of tumor lysis, they had an effector to target ratio of 1. They had as many CAR cells as they had T cells. And the way that happened was there was massive proliferation of the CAR cells in the patient. They literally became bioreactors. The patients grow most of the cells. We don't do it in the lab. It happens in the patients. So there's 2 ways to get huge numbers of cells. One is to grow up large numbers in vats in a lab with TIL therapy. And the other way is to infuse small numbers of cells, which we did, and then let them proliferate and engineer them to proliferate, which is what a CAR domain can do. So this is Bill Ludwig's PK. This is a semi-log scale out to a year here on this x-axis. And this is the number of CARs per microgram of genomic DNA in blood. And it's very similar to what we found in bone marrow. And he had a massive proliferation, cytokine release syndrome, and then These are CAR memory cells, and they now persist for at least 8 years. We had a 57% overall response rate in the phase 1 trial, and if you arrayed the CR patients, the partial responders, and the non-responders, you can see there's a big difference in the PK. So these patients, the cells barely proliferate, and in every tumor we've looked at, proliferation of the CAR cells correlates with response. So that's the biomarker we want to optimize. And these non-responding patients, what we found is there's a cell intrinsic defect in their T cells. It looks like they're exhausted to start with, and then they don't proliferate when you infuse them, and then they don't respond. So we then have now analyzed these patients with retrospectively, and because we can continue to follow the CAR cells in the patient, because the CAR is expressed on the surface of their cells, and genetically we can find the integrated DNA. And David Porter was the first PI, and then he did this work with Joost Mellenhorst in Nature Medicine last year. They PK again for patients 1 and 2 that both have ongoing CRs as shown there. And this is the number of normal B cells they have, which is 0. They have germline, they have complete absence of B cells, a B cell aplasia, and which tells us the CARs are still working, because if they lose their CARs, the B cells come back. So this is the first, really living drug. We gave them 1 infusion, and now it's lasting a very long time. So it's very different than the standard medical practice of recursive administration of drugs. And so it's more like a vaccine, where you can have a lifelong effect. So we characterize those cells, and they look like memory CAR cells, and all the details are in this Nature Medicine paper of what they look like. We find them in the bone marrow and the blood. Okay, so going back to the PK of these CLL patients, patient 10 turned out to be really very interesting. He has a delayed peak, which you can see here. It took 50 days after a second infusion until he had this peak here, the Cmax. This was 10 days here in Bill Ludwig. So we asked why that was, and so this is the patient. He had 2 infusions. The first time nothing happened, and he continued to have circulating leukemia cells, which are shown here, 10,000 leukemia cells per mil, a lot of blasts circulating. We infused him again, and then he had cytokine release syndrome. This is interferon gamma and IL-6. And his leukemia cells went away. He's now 4 years after this, and he's in a complete remission. He was 77 years old at the time. So we asked, why did he have this really late response? He had massive periabdominal, periaortic masses, paramediastinal masses, and so on, and it all went away. And he had a delayed tumor lysis syndrome and macrophage activation syndrome that required treatment for cytokine release syndrome. Turned out his CAR had integrated into a gene called TET2. And TET enzymes work on epigenetics. So they have a dioxygenase gene that takes the DNA methylcytosine and converts it to hydroxymethylcytosine. That's what TETs do. So they change your epigenetic landscape and make it change expression of genes. And so there are 3 TET genes, but we found this patient had a TET2 integration, and he had clonal T cell proliferation at the time of his tumor lysis syndrome. So these are patients 1 and 2. They had many different T cell receptors at, where they had their CARs. This patient, it was only 1 T cell. There, we could say he had a V beta 5.1 T cell, CD8 positive, and so that contained all of his CARs. So they were all progeny of 1 cell. And when we looked at the infusion product, we use a lentiviral vector, and we transduce CD4 cells and CD8 cells. And there were over 1,045 unique integration sites we could find. So that's what went into his arm. But at the time of tumor lysis syndrome, there was only 1 integration, and it was by this heat map. It was TET2. So a very interesting evolution of a polyclonal product to one outran all the rest. And it's because of a gain of function by losing TET2 function. So we found when we analyzed his CAR cells that there was a single copy of the CAR in chromosome 424, and it introduced a loss of function by making premature stop codons. So TET2 has its dioxygenase, where it takes off methylcytosine, down here in exon 11, and their integrated CAR was right here. So this part never gets transcribed, and that's the mechanism of how he had loss of function, a lucky integration into that gene. So we found that it reprogrammed his CAR cells, so we could actually take his CAR T cells and compare them to the non-CAR cells, which are great controls. And we found that these transcription factor motifs were gained. So epigenetic reprogramming, these are good things to have for effective CAR T cells compared to his other non-CAR T cells. By gene ontogeny, we found things, these are very high p-values of difference in his CAR cell compared to his non-CAR cells, and they correlate with cell cycle progression and healthy T cells. And there was differences epigenetically in loci, and this just showed interferon gamma, for instance. So there's reprogramming of these cells. So we asked then, could we do this and phenocopy this in normal T cells from a healthy donor instead of a patient who had leukemia? So we used siRNA knockdown technology, and it's all described in this Nature paper last year. But what happens is if you add leukemia cells then to cells that have knocked down TET2, after a couple of weeks, they start proliferating a lot more, and they have restrained differentiation. They last longer and work better. So it's a really important example of forward and reverse genetics that happen in this paper. By following the cells in the patient, we found that people often ask, what's a dose of a CAR T cell you need to give if it's reproducing, if it divides in the patient? The answer is it could be one. That's what we found in this patient. The progeny of one cell cured the guy. So if we could do this reproducibly and it's safe, then the manufacturing costs go way down. So the lowest effective dose could be as low as one. Unintentional knockdown and knockout of TET2 enhances CAR function. So we're looking at doing this now prospectively to see, especially in old people where their immune systems don't work as well. Kids don't really need this. Their immune systems are much more potent. So that's an example of you can follow cells long term. You can find out whether they work or they don't work because they're genetically marked. So then that was CLL and lymphomas turn into acute lymphoblastic lymphoma and leukemia. It's a really awful disease. So if you have relapsed leukemia, the data from Germany, their multicenter ALL group, with more than 2,000 patients, they die at the same rate as pancreatic cancer. Less than 10% of them survive 2 years. And so those are the patients we put on our trials beginning in 2012, acute leukemia, adults and kids. And our first pediatric patient is shown here, which was Emily Whitehead. And at the time we treated her, her bone marrow showed 99% of the cells in her bone marrow were all one clone, leukemic. And at 6 days after treatment, it was down to about 50% by staining for CD19 and 20. And she had very few CARs in the marrow that day, but 17 days later, she had 3/4 of the cells, T cells in her bone marrow were CAR T cells, and no more leukemia. In 17 days, pounds of leukemia went away in her. And CR now is more than 7 years in duration. So she not only met the Pope, as David showed you, this is with her on January 31st in 2015. So President Obama called her to the White House to announce the initiation of a $250 million precision medicine initiative. And he used her as the example. And she was in the front row. And afterwards, they have a receiving line. And her father, Tom Whitehead, who I see very frequently, said, the Pope, Obama asked, is there anything I could do for you? And Obama and Tom Whitehead said, please excuse, she needs a reason. It was a Monday, so it was a Monday, and literally he wrote this excuse on White House stationery, excuse her, she was with me. And so this is just in the Philadelphia Inquirer now, a few years later this spring, and it's amazing, 7 years. And so they're making a full-length documentary about her and the story. So, okay, this is Emily, though, in the New England Journal. Now back to the medical part of it. So she had almost lethal cytokine release syndrome. This is her fever curve. She was the first patient, and we had no idea that the childhood immune system was more potent than old people with chronic leukemia. And so she had 106-degree fever for 3 days. It was not infectious, and it was refractory to high-dose steroids and TNF blockade after we found that she was not infected. And then we found she had very high, 1,000-fold above normal levels of IL-6. And so we gave her an IL-6 blocking antibody, tocilizumab, that blocks the receptor, and it immediately made her fever go away. And she went from being do not resuscitate and on a ventilator to waking up. And so the FDA did a very unusual thing. They co-labeled tocilizumab, a drug made for arthritis, with the CARs that Gilead and Novartis made. So it's co-labeled to treat, but it's FDA initial approvals for JRA and adult RA and Castleman's in Japan. And there's a lot of experimental therapies going on for cytokine release syndrome. We found that it's actually a good thing to have. The non-responding patients don't get cytokine release syndrome. So it's an on-target effect. Unlike when you get chemotherapy and you get mucositis and so on, those are off-target effects and are not related to you getting better. So cytokine release syndrome happens. It happens more frequently and is more severe in patients with high tumor burdens. And so if you can treat people up front at lower tumor burdens, it's an outpatient therapy. But when you treat late-stage patients like we did in our initial trials, about half of them require intensive care therapy. But it's almost always reversible. This is the trial we did in Philadelphia. Were the first 60 patients, and it was incredible. So there was a 90% CR rate. The first one out here, this shows out to 30 months. That's Emily, and so a very high rate of complete remissions. And basically, the only relapses have been due to where the leukemia loses its target molecule, CD19, which in a way kind of proves the mechanism as CD19 directed. So this is, again, looking at Emily and the second patient we treated, the PK in the blood, and it peaked 7 days after we treated them, very high level. This is again, copies of the CAR per microgram DNA. When you back calculate, this is essentially 100% of the blood cells are CAR cells at that peak. And then and they persisted now for 7 years in Emily. And you can do a right stain at the time of peak, and this is what these CAR cells look like. They're very activated lymphocytes. And a big surprise came when we did lumbar punctures, and we found in all our patients that, and this is at a month, that there were CARs in the cerebral spinal fluid, even though they had no leukemia. And you can do a cytospin, and this is what the CARs look like there. So they travel around the body to all areas we've looked at. There are side effects. I mentioned cytokine release syndrome. There's a potential for insertional oncogenesis by the lentiviral vector. So far, that's not happened. No one has, with more than 1,000 patients treated, no one's gotten a T-cell leukemia. And then there's an unexplained syndrome of neurologic toxicity, which usually follows the cytokine release syndrome temporally and probably has several mechanisms of action. And we've been studying that. And this is work that's still unpublished, but done by Avery Posey from the University of Pennsylvania and Howard Chang at Stanford. So Howard is making a map of the mouse brain on a single-cell level of all the transcript using single-cell RNA-seq and ATAC-seq to map the brain. And what he came up to me and said, I found that there's CD19 in the mouse brain, and which is a complete surprise. If you look at an immunology textbook, CD19 only comes on in B cells through PAX5, a transcription factor turns on, and the cell either becomes a B cell or a T cell. So that's 30 years worth of research. And by single cell assays, this is what he finds. Parasites on these Tisney plots express as a canonical parasite molecule, CD248. They express CD19, but they don't express other B cell molecules. Like this is a B cell receptor. They don't express CD20. They're very unique this way. And he's found it both in human parasites and mouse parasites, but only those in the brain, not in the parasites that line the blood vessels in the arms. So there's some unknown function there and unexpected PAX5 independent expression of CD19 in the brain. And this just shows more of that in violent plots. If you look at CD19, parasites are way compared to the rest of the cells in neurons, for instance, in the brain. And you can see it by IHC. So it's been missed for 30 years. It's rare cells. But when you do careful IHC, you can see it. And neuropathologists say these look like parasites. And they stain with anti-CD19, in this case, in the hippocampus. So does it have anything to do with neurotoxicity? So Avery Posey made a model to test that in mice, and he made an anti-mouse CD19 CAR, and he made one that has a 4-1BB signaling domain or one that has a CD28 signaling domain. And as a control, they made one that's, again, it's a human CD19. It doesn't bind to mouse. And what he found in both immunocompetent mice and incompetent NSG mice, given these T cells, he looked at Evans blue dye just at mouse brains a few days after giving them CAR cells, and he could find in both kinds of mice, if they had the CD28-based CAR that targeted mouse CD19 but not the others, a lot of Evans blue dye. And in fact, as much as if you gave mannitol, which is what classically, neuroscientists use to break down a blood-brain barrier. And this just shows more of that by imaging using gadolinium. So it's dependent on the signaling domain and the targeting of CD19. And it doesn't say that's the only mechanism, but it's probably one. So what about the PK then of this living drug? We worked with some classic pharmacologists at Novartis to model this using compartmental analysis, looking at a variety of factors that are described in these papers. And there's an expansion phase that reaches a Cmax, and then a contraction. This has a doubling time in the blood of 0.7 days. So the CAR T cells very rapidly accumulate, and then they have an initial half-life of 4 days, and then an essentially infinite half-life in about 3/4 of the patients where there's long-term engraftment. And we have not found any relationship between the dose of the CAR cells and Cmax. And we think that's because they're proliferating. The Cmax really depends more on how much target there is in the patient. So what's their burden of leukemia? So it's very different to have a proliferative drug, from the pharmacologic persistence aspect than one that dies away. So CAR T cells are living drugs. They can evolve in patients. They can traffic and persist and stay on immunosurveillance. We know that autologous T cells are very safe. They've been given by transplanters for decades, and they have never transformed. And even with genetic modification, they have not. Now, that's not the same with stem cells. Hematopoietic stem cells are more susceptible to being transformed and becoming leukemia cells. So there's a different safety factor depending on what kind of cell that you genetically modify. But post-thymic T cells are very resistant to transforming. I mean, we've tried and failed. So this is some data where from Bruce Levine, where we've done in our manufacturing center, treated, in this case, 476 patients with many kinds of engineered T cells, including for HIV and then different cancer trials, using various vectors and so on. And we, because of FDA policy, have to follow the patients for 15 years after they're treated. And so we have over now 1,700 patient years of safety and not one case where they've transformed. Now, if you give patients leukemia, who have breast or ovarian cancer and you give them chemotherapy, about 5% of the survivors will get secondary AML, leukemia. So chemotherapy causes at a fairly high frequency stem cell damage, and then later you get AML. So far, T cells have a much lower risk of that than chemotherapy, but it will eventually happen, I'm sure. So, okay, we have a number of trials underway now at Penn, that are moving beyond leukemia. Some using engineered T cell receptors. This is for brain cancer, EGFR variant 3. Avery Posey is just opened a trial targeting a glycan called TNMUC1. And I'm going to describe a little bit about a trial called targeting FAP and PSMA, prostate-specific membrane antigen for prostate cancer. So the limitations right now, we haven't had home runs in solid tumors yet with engineered T cells. That's the central issue of the field. One reason probably are intrinsic T cell limitations such as exhaustion. The cells get, and there could be extrinsic things, maybe the immune system rejects the CAR T cells, or the tumor microenvironment kills the CAR T cells, or the blood vessels or stroma and so on don't allow penetration. So these things are much more complex than they are in the case of hematologic malignancies, where there are much less mutations in general than there are in solid tumors. Several investigators, Rainer Brenchens at Memorial Sloan Kettering calls them armored cars, and Henrik Apken now in Germany calls them trucks, but making more other modifications rather than just a single car that I showed you for CD19. And these do many different potential things, enhancing proliferation. We're just opening a trial where the car makes IL-18, which will activate the natural innate immune system, and so on. And so I think those will be in the field. That's what the field's moving to for solid tumors. And we have an armored car now for prostate cancer, making a dominant negative receptor for TGF-beta, transforming growth factor beta. So prostate cancer is the most common cancer in men, and most adenocarcinoma of the prostate expresses PSMA. It's an extracellular enzyme that has roles in the metabolism of glutamate and folate. And PSMA actually increases on the surface of prostate cancer cells with advancing stage and aggressiveness. And TGF-beta is actually made by prostate cancer cells, and it's immunosuppressive, and it actually promotes tumor cell stroma and tumor cell growth. So it's now viewed as a checkpoint, much like PD-1 and CTLA-4. If you would block that, you could enhance potentially the immune system. So Chris Kloss in my lab made this armored car that has a dominant negative receptor to TGF-beta and targets PSMA using a lentiviral vector system. And so he found, really quite surprisingly, that these CAR T cells are resistant to exhaustion. And the main experiments shown here where the cells over a period of 6 weeks got addition of prostate cancer cells, and then, to an initial seeding of a petri dish that had CAR T cells in it. And if they're, control CAR cells are CD19 specific, they just die away. But if they're prostate cancer cells that express PSMA, then the CAR T cells that are PSMA specific divide, but they stop dividing after about 21 days if they're wild type CAR cells and have intact TGF-beta signaling. But if they have the dominant negative receptor, they go for 3 or 4 weeks more, proliferating and still killing tumor cells. So that's the rationale to start our trial. And it's a phase 1 trial ongoing. We've treated now 8 patients at the University of Pennsylvania. It's led by Naomi Haas and Vivek Narayan. And we started initially with a cohort that got 10 to the 7th CARs per meter squared, and then cohort 2, 10 to the 8th per meter squared, and then we have one patient treated where we gave lympho-depleting Cytokine chemotherapy and then the CARs. So no previous chemotherapy in these cases. And these are patients that have, castrate-resistant, hormone-resistant on multiple lines of therapy. And the first 3 patients we treated are shown here, the PK. And what I can say is this is much better than our other solid tumor, like we've reported in brain cancer and pancreatic cancer, CARs targeting different targets, but we haven't seen this good of proliferation, and it happens around the same peak as we see in leukemia patients. And in December, we treated this patient who had, refractory prostate cancer after 4 lines of standard therapy. So he was the first one to get preceding chemotherapy to make lymphodepletion, which helps the CAR T cells proliferate. And this is a non-linear x-axis with preceding 4 months, he is having exponentially increasing PSA. We gave the CAR cells, and it went to undetectable levels in 3 weeks. So it went from 35 to less than 1. And he also had very severe cytokine release syndrome and required grade 4 CRS, required multiple interventions for that. This is ferritin, which is a marker of cytokine release syndrome. So this is the first time in a solid tumor we've seen really, it looks very much like what we see in leukemia, and we're excited about that. I've graphed here the PK of these patients on this ongoing phase 1 trial. Blue is the lowest dose cohort, red's the second, and this is the third. And what's exciting about that is if you look at the log copies per microgram of the CAR in blood, it's the same as what we see pretty much in leukemia. Now here, 90% of the patients got CR, and the patients who don't get a CR, they're on the low end here. So proliferation and Cmax matters, and we think this has really enhanced this dominant negative receptor here in prostate cancer. So what I've showed you is, targeting the tumor cell, in this case PSMA. This is work with Steve Albelda and Ellen Pire, where we're also looking at targeting tumor stroma. And then in the end, we want to target both the stroma and the tumor. So the first approach to that is that we're going to do is target cancer or carcinoma-associated fibroblasts, or CAFs, as they're often called. And there's an extensive literature in pancreatic cancer and other tumors that they have many things to promote the growth of the tumor and suppress the immune system. And targeting them may impair the ability of this tumor to thrive, is the hypothesis here. And so if you look at pancreatic cancer and stain with cytokeratin, that's the tumor. And if you stain instead for FAP, which is fibroblast activation protein, it's a serine protease, it's not on the tumor, but it's on the surrounding cells. So that's what we're targeting in this case, CAFs. And it's in many. In this case, it's lung cancer, but it's in would look just like that in pancreatic cancer. And this shows more of that, say, lung cancer H&E. Here's FAP, again, surrounding tumor islets. And here's the EpCAM staining the tumor. So same thing for ovarian cancer. So it's a dipeptidylpeptidase. It's overexpressed on these fibroblasts. It's not on healthy fibroblasts. And so we made a CAR against that, and again, having the same signaling domain, and we reported this in 2014, that we could treat mouse tumors with an anti-FAP CAR. This is just some examples of different tumor lines treated. Either SS1 is an anti-mesothelin, and the mesothelin negative tumor doesn't respond, but the FAP is in green here very effectively, and in this case it's FAP CAR here compared to a non-transduced control, and you can see here FAP compared to a mesothelin CAR. So they have very potent effects. And we have a phase 1 trial just beginning later this year on this. So unfortunately, we don't have any clinical data yet, but we're hoping that we'll be able to establish safety and then find on tumor biopsies if we've eradicated the cells that express FAP would be the primary endpoint that we want to be able to see. But more recently, I'm right under a floor, which is developmental biology, and they knew we were making FAP CARs, and they came to us and said, we find FAP on their failing hearts. And they said, what about if we would treat fibrosis, which is a really big problem. Once you get scar down, it's been pretty much said to be irreversible. Could we fix that? So this is work from John Epstein's lab at Penn, and where all we did was make the CARs. So this is human staining from John's lab of hypertrophic and dilated cardiomyopathies, and green is FAP. So humans express FAP in sick hearts, but not healthy hearts. And so that's the hypothesis we want to test in a mouse model. Could we target that? And so the way that was done with Haig Akajanian, who's a postdoc, is to get mice and then treat them with angiotensin and phenylephrine, which is a stress to the heart. Within a month, they have cardiomyopathy. And we gave injections of FAP CARs or control CARs to these mice under these conditions. And this is what we found. So the healthy heart does not have FAP staining, but a mouse treated with angiotensin and phenylephrine who has cardiomyopathy, you can see lots of fibrosis here. And if they got the FAP CARs, it's really, even though they had the stress, they don't have it. And this is a quantitative assay of that the FAP CAR could, given after the start of this is a week to 2 weeks after, could regenerate this. And so that looks as a promising thing, beyond and outside of cancer in fibrosing conditions, perhaps in autoimmune disease. So there are a number of therapies that we'll see now in oncology where we'll combine CAR cells. We have trials right now with combining with checkpoint therapies, for instance, in CAR cells, with radiation, small molecules like ibrutinib and CAR cells, vaccines, oncolytic viruses, stem cell transplants. And, so for different tumors, there's different sort of low-hanging fruit right now that can be tested. So I've shown a lot of data today about CAR cells having potent activity in hematologic malignancies, and some activity in solid tumors, and that's the major research in the field. There's off-target and on-target toxicity with CAR cells that needs to be dealt with, much as in bone marrow transplantation, where the major limitation, has been graft versus host disease. So in the case of CAR cells, it's not GVH, it's CRS. And so neurotoxicity may be related, at least in part, to parasite expression. And there's a lot of things that affect how a CAR works. It's, how it's designed in the signaling domains, the binding affinities, the cell subset that are expanded, and how they're cultured. And a lot of room now for further optimization. So this is my CAR lab. Joe Frietta is a new assistant professor, but he's a postdoc. He's the one who discovered the TET2 functions that I showed. And a number of people involved at the University of Pennsylvania. Bruce Levine, my long-term colleague since 1994, has done all the cell manufacturing. Steve Alberto is leading the FAP-CAR work. Steve grew up the pediatric ALL work. And our colleagues at Novartis, who translated this from a small academic lab to a global therapy. So thank you very much for your attention.